this evening. Ja, ich freue mich sehr, dass Sie alle gekommen sind. Welcome everybody. This will be an English spoken evening actually. And it is my pleasure to introduce to you Angelika Hilbeck, with whom I have worked since a decade now, I think, or so. And whenever
probably a bit more, I'm afraid.
Whenever things get really complicated, I call Angelika and ask her, how does this work? Angelika will present to us what we call in German eine steile These. A really provocative thesis about the whole issue of GMOs and the question of whether this science is probably something like the emperor's new clothes.
Perhaps.
We'll leave this
we will discuss that
to
exactly.
Angelika, and she will also introduce to you, after her short presentation, the rest of the panel, and I will try to keep the audience calm until until we have the opportunity to discuss it then with all of them. You'll start?
Okay. Shall I say a little who I am?
Maybe
other than your consultant on
exactly.
biology.
Maybe. Yeah.
Well, you see some of that here. I'm the co-founder and a board member of this science group we have created in 2009. Next year we will celebrate the first decade of our existence, the European Network of Scientists for Social and Environmental Responsibility. But that doesn't pay my rent, the work for that. So my rent I pay with my work at the Swiss Federal Institute of Technology, where I am a researcher and a lecturer in Switzerland, in Zurich, on environmental biosafety and agroecology. So they pay me, and here I am now today with my hat as a co-founder and a board member of this network. And because we're going to make a bit of a provocative thesis, as he said already, and I rather do this under this hat than under my science head to protect us a little bit. All right, so you see it in the title already, Promissory Science Bound to Fail. That's already a bit of a provocation right there. But actually it's not that provocative, because if I look at this poster here that was just put up there, pretty much what I'm going to say, or a lot of it, is already here. So other people have been making these suggestions as well. We want to study and analyze and discuss with you and with our experts we have here, have a debate about the scientific foundations of genetic engineering, something I feel and many of us feel we should have actually done at least 20 years ago when we started on this adventure, or maybe even 30 and 40 years ago when the necessary tools, the molecular scissors, were discovered and we started on this journey of engineering organisms and engineering DNA. All of you, I guess, who are in this room have heard about what has been promised right from the get-go. In the 70s, early 70s is when they discovered the first molecular scissors, and right from there on, the sky was the limit when it came to what you could be doing with these tools. But today, 30, 40 years later, 20 years, more than 20 years down the road after the first commercial GMOs were introduced into the agro and food system, we're missing products. Why has it not delivered on the promises that were made? And more the question, will it ever deliver? Or are there lessons to be learned that might also inform what we could expect from the new scissors that are now coming along, and where we hear new promises? So these are some of the promises. I'm not going to go into. This is just about what was done in the green, what we call green gene technology for the agro-food system. Similar promises have been made also for the medical field, but that's not my field, so I stay away from that. But for food and agriculture, this is what was promised: abolishing hunger and malnutrition, higher yields, adapting the plants to the changing climate, introduce tolerances against pests, diseases, viruses, what have you, introduce better nutritional value, vitamins, iron fortification, what have you, was all promised. And this was the way it was envisioned it could happen. And do not blame me for this presentation. I'm just the messenger. The people who've put this out is the agrobiotechnology industry, This is taken straight from the website of ISAAA, a platform of the biotech industry, and where they have put up their vision how they think they can make, for example, golden rice. A 4-step process is all it takes. Actually, it is the same vision that underpins the making of any GMO, the idea, the concept that was behind it. And it was a 4-step process, not only to feed the poor, but to feed anybody and create any plant. And 3 of these steps were going to take place very simply already or only in the petri dish, only in the laboratory. And it would start with identifying a donor organism where you have a trait, a phenotype that you want to take the trait from and put it in another organism that is unrelated to the donor organism. So you have to identify the DNA, the nucleotide sequence on where you think this trait is coded. You cut it out with these scissors. You put it on a shuttle, on a ballistic shuttle or on an agrobacteria shuttle, and you put it into your recipient plant. So 3 of the 4 steps happen right in the petri dish. And it was thought that from there, it would be just an easy 2nd or 4th step, 1 small step, to put it in the plant, and then out you go, and you grow it in the field. That's the idea behind it. And you feed, not only do you grow it in the field, but you feed the poor according to them. So based on these ideas, on these conceptual ideas, how simply this process should be, these ideas came in the early 90s. These were the kinds of presentations that I saw personally, given at my university in the United States, where I did my PhD. In the early 90s, this is a slide I could find on the internet. This is pre-internet days, so you don't find many of these presentations online where you can say, look, this is what I've been presented, I've been given, I've been told in the early 90s or late 80s, how this field would develop and how it would go in leaps. In 5-year leaps, as you say, we would be leaping to another level. We would be out of agronomic traits by the year 2000. I heard countless presentations at my university in the US where this was just a given. It was not even questioned, that's how it would be. Agronomic traits would be herbicide resistance, insect resistance, that kind of thing. We would be leaping then. Why it would be leaping, I don't know either. The scientific foundations of the leaps, I've never been explained. But we would be leaping into food processing traits called output traits, or customer traits, consumer traits, so that you would be getting the better food, more adapted, better oil compositions, starch compositions. Then we would be going into pharmaceuticals or cosmeceuticals or other kinds of industrial products, specialty chemicals, and we're over here now, so we're way off this. And it was promised to deliver. So it was not maybe we try and perhaps it works, that is our vision. No, it was like this is what we will be doing. This is a promise. Now the reality today, 2018, so way down the road from these promises, is we have plants, 99% of all of the GM crops grown in the field have 2 traits. Herbicide resistance, vast majority of them based on glyphosate-based herbicides against these, and insect resistance, all of them based out of 1 bacteria, Bacillus thuringiensis. What we see in innovation coming along is that these 2 traits are being combined through conventional breeding, notably, less so through molecular stacking. And the golden rice still doesn't exist to this day, because that small step that was envisioned to produce a golden rice into a plant, exactly this step has not worked today. All the attempts and tries they have done to put that golden gene trait into a rice variety that would be grown by farmers have so far failed. They have not delivered plants that would be acceptable to farmers and would function in a market and in a market environment and in their actual production environment. By higher yields was something that was also promised. By 2009, we heard from the United States, one of the heartlands where they grow GM crops, that the promise, contrary to the myths about superiority of GE crop yields, the yield gains that were observable, yields have always been increasing, but they were not due to the GMOs, they were due to traditional breeding and other improvements in the cultivation of these crops. 2013, our colleague who will be debating with us here, Jack Heinemann, tonight, and his colleagues have done a comparison of yield data that are submitted to the FAO, the World Food Organization, and have compared how two comparable regions in terms of production and access to the means of production have fared, and how the yields have developed in the region that did adopt GMOs and the region that did not adopt the GMOs, and they virtually didn't find any difference for the crops that both regions were growing. By 2016, the New York Times came up and checked and completed the data and said, has anything changed since then? And they found no, nothing has changed. That line continues to increase, but there was no difference to be seen that those regions who would grow GM crops would have a steeper increase in yields. So yield gains wasn't delivered. The New York Times spoke of broken promises because they were promised, and these promises were broken. Why was that? The same with drought-tolerant plants or plants adapted to difficult environments. There is one GM trait, or one crop out today by Monsanto, where Monsanto claims that they have a trait incorporated that improves what they call the yield protection under moderate water limited conditions. So it's a very complicated phrasing they use. But if you go and look in the Web of Science to the scientific literature, you don't find any independent peer-reviewed literature that actually looked whether that is true or not. So that today is still unconfirmed, and you hardly hear anything about this so-called drought-tolerant maize. That also occurred to Nature in 2014 when they said crossbreeding crops is actually delivering. We do have drought-tolerant maize varieties. We have very good ones, actually. I work with them. They have been produced through normal, conventional breeding, sometimes with the use of molecular breeding, of marker-assisted breeding, but not GM. So these promises made 2 decades ago have really not materialized. We are still stuck in the agronomic trade phase. We have never left it for over 20 years. And somebody must ask the question, why is that? Now, with that not having delivered, a new kid comes up on the block, and that's called gene editing, CRISPR-Cas. And instead of trying to work out why the first generation of genetic engineering tools hasn't quite delivered and worked, so that maybe there could be lessons to be learned for the next generation, they're coming with the same promises, just bigger, even bigger if that's possible. A DNA revolution has been, again, promised by National Geographic, a modern Prometheus using editing the human genome, and a very well-known weekly newspaper of Germany has been glorifying. I mean, this has taken on really a bit of a religious touch even already, with this image there, glorifying and speaking of the grand hope. So finally we would be dealing with all the ailments of humans, and finally we would reach what we haven't reached with the first, when they made the exact same claims. No critical debate, no critical analysis why and what is the reason and what is the science behind it that hasn't delivered in the first time. Again, also 2014, in the realm of CRISPR-Cas, a Newsweek article was saying GMO scientists could save the world from hunger if we just let them. Really? Again, trying to save the world with point mutations? Have we not learned from the last 20 years where we tried also that and it hasn't worked? So why? That is our question. The other side, the biotech side, has identified, it hasn't escaped them that they haven't delivered. But their analysis, why that is somewhat different from ours. They put it squarely on regulations. The regulations are the obstacle and they are blocking progress. But there is no regulation that would stop anybody in North and South America. South Africa neither, but mainly in North and South America. North America blames or boasts that they are deregulating plants. They don't have a regulatory system. You can do what you want there. Why haven't they delivered there? There is nobody who stops them. And a good colleague from my university was also blaming fully squarely on the regulation that must be revolutionized, which really means do away with them, that unjustified and impractical legal requirements are stopping genetically engineered crops from saving millions from starvation and malnutrition. Really? That is our question. Or is it maybe because it's based on outdated science. Maybe they haven't done the right analysis and haven't learned from what other fields in genetics have been finding over the years. When they found and discovered the DNA as the material and structure that carried the genetic information, they also came across with a dogma regarding its function and its form. And that dogma, you know, everybody knows what a dogma means, was not to be questioned. And it held the idea that only information is going to be passed on to the next generation that is coded in DNA, in nucleotide sequences, and nothing else. And it will only go in one direction. And nucleotides were being likened to a digital code, that you could use those 4 nucleotides and replace the binary code of the IT field with it. And if that would work, that would really be a quantum leap indeed. So it was early on, this is also a paper from Nature in 2003, where they reported that the remarkable feature of the structure of DNA is that it can accommodate almost any sequence of base pairs, and hence convey any digital message or information. And that it has 2 types of digital information you can manipulate, and you can construct genes that encode protein and genes that encode regulatory networks that determine the function of it. And they came, again, the discovery with these kinds of statements that the discovery of DNA started us on a journey. At its end will be the grand unification of the biological sciences and the emerging information-based view of biology and of life. Really, life as a construction kit where you just replace binary code with the 4-digit of the DNA, and then these kinds of imaginaries would come true, that you build your molecular computer, and terminology like intentional biology, digital biology has been coming across and has been attracting an enormous amount of money. These fields, these are not these are fantasy fields. We will decide and we will discuss it, but they have been attracting huge amounts of money. Venture capital is still going in there at a massive amount. But there was one problem they always had, and if you read those papers, they make good reading to understand how they see the world and how they view it. It's when they take these organisms or the pathways they generate in the lab out from the highly controlled, very stable and steady conditions of the lab into the real world, into the world where you have fluctuating conditions. Then the environment comes in and they have noise. They have these stochastic information or the stochastic events that happen depending on what the environmental conditions is, and they generate noise. So noise that ruins their signals, ruins their idea, and that need to be attenuated. And so their entire energy goes on how can we control this noise and how can we control and get these organisms to function like computers. Now what they're missing really is the other field in science that has been seeing an enormous input these days, and a lot new knowledge has been coming out, is what's called epigenetics. We might hear something about it, how you view it, because the term epigenetics already involves a gene in it. But really what it says is it's information passed on to the next generation that is not coded in a DNA. So that good old central dogma has been basically falsified right there. You can convey information to the next generation based on your experience, what you do in your life and what you experience in your life. You can pass on without changing your DNA. That is pretty revolutionary, that's pretty fundamental, but it has not come into the field of biotechnology. And this is what astounds us, what surprises us. How is that possible? And these are my last 3 slides where I'd like to get you guys then also thinking. For me, this is how the field presents itself. We have a timeline where we sort of co-discover things. We co-discover how it works. It's the biology, the DNA was discovered. A dogma was created, a narrative around it. Then we went into discovering scissors where we could, manipulate DNA and see what happens with the organisms. And then came the big surprise in 2001 as our capacity to sequence DNA and to really get the sequence of all of these nucleotides, when they discovered, after sequencing the human genome, that humans have less genes than most plants. That we don't have more protein coding genes than a tiny little nematode, for example, only minor. And nobody would claim that a tiny little nematode is as complex as a human, or a human as simple as a nematode. So something wasn't quite working out. It didn't line up with what we thought how it would work and what we saw how it worked. But instead of then going and inquiring more, what I observe, and this is something I'd like to discuss, is like the field really split up, very much so, into those who pushed for more and more engineering. That's when all these ideas that I just presented to you came up in the early 2000s, right after the discovery of the human genome, and that the genetics as we thought it would work was shown wouldn't work. You saw the push into the genetic engineering, into the synthetic biology, where it got more and more narrow. While in the other field, in the epigenetics field, finally got the recognition it deserved. Epigenetic study is not young. It was also already decades ago discovered and seen by researchers that the idea of this linear functioning and this DNA-based functioning wouldn't quite line up with other observations. But they were minority, they were marginalized, they weren't listened to. But meanwhile, also in the early 2000s, they started to gain recognition that there is validity to their findings. And the field is sort of getting broader and broader, to the point where today, and we've been discussing this often, the concept, gene as a concept is becoming blurry. Does it even exist, that gene? The RNA world was discovered, the epigenetics world was discovered, and yet it didn't permeate in the other part of the science. There is a wall where you just can't go through. And what became clearer and clearer in the ecological genetics, that issues, traits like variability, plasticity, adaptability of an organism with emergent properties is the key to survival and to evolution. It became on the other side of the wall. It became noise that needed to be quenched, that needed to be attenuated. So what I see is a real schism in biology. That's how it presents it to me, with one having fantasies of making life into machines, intentional digital biology, and the other saying, I'm not even sure genes exist. I'm not even sure about a lot of things anymore that we thought 20 years ago we had figured out. So that's what we want to discuss. Let's debate the dogma. 3 questions I wanted to put to you. You can choose how you wish to address them. These are the scientific limits to gene technology as I see them. Separate the science from the science fiction. What kind of predictions can we solidly make? What does the science support, and from where on is it more fiction than science? And has this whole endeavor of biotechnology become, meanwhile, with all the enormous funding that went into it, too big to fail, so that we cannot take that step back anymore and critically reflect, because the consequences of having to admit that we were putting our money into a bound to fail exercise is just too big to swallow. And the business concepts behind it, the money that went into it, and how and if should the civil society start to build in these understandings of the limits of gene technology, the scientific limits of it, before we start dealing with doing science fiction risk assessment of science fiction organisms. So that's what I'd like to discuss with you guys, and it was my job to introduce them, right? So I'll do that.
Can we clap?